Good morning and good afternoon, everyone, and welcome to our today's webinar. We are really glad to have you all here. My name is Valentina Ratti, and I will be your host for this session. I am pleased to introduce our speaker today, Claudio Maestri. Claudio is our product manager at Fedegari, with more than 15 years of experience tackling the technical challenges in the pharmaceutical industry. His mission is to help organizations to move beyond outdated sterilization methods. In an industry where patient safety is at risk and poor choices can have severe consequences, Claudio guides professionals towards safer, more effective alternatives, making the transition beyond EtO, as in this case, an imperative choice. But before we get started, just a quick note about today's session. If you experience any technical issue, for example, audio or connection problems, please use the chat to let us know. For any questions related to the presentation, we kindly ask you to use the Q&A button instead. We will address all of your questions at the end of the presentation during the Q&A session. Okay, and now let's get started. Claudio, the floor is yours.
Thank you, Valentina. Good morning, everyone, and thank you for being here. Today, we are going to explore the future of sterilization technologies, specifically why vaporized hydrogen peroxide and saturated steam are shaping up as the most reliable alternatives to ethylene oxide. For decades, EtO has been the backbone of medical devices sterilization, but things are changing and fast. On those changes have real consequences for manufacturer, regulators, and most importantly, for patients. Special attention is given to the challenge associated with the new modalities and how advanced technique can address critical issues like residue management and sterile penetration. While moist heat sterilization remains a preferred option for compatible materials, vaporized hydrogen peroxide has gained significant traction as a viable low-temperature alternative for a broad range of sensitive devices, especially with its recent FDA Category I classification. So let me take a moment to present today's agenda. We will start with what sterilization by design means and why is the Fedegari identity. A quick overview of the regulation and guidelines behind this transition from EtO to alternative sterilization method. And then H2 advantages and challenge and how the deep vacuum is the solution to win this challenge. EtO and H2 comparison, saturated steam and vaporized hydrogen peroxide as alternative solution to ethylene oxide, and at the end, some case studies like saturated steam sterilization of ready-to-fill primary containers, H2 deep vacuum sterilization of hemodialysis blood tubing sets, H2 deep vacuum sterilization of glass per-fill syringe. So today, Fedegari is a global leader in providing highly customized integrated solution for the biotechnological and pharmaceutical industries, with several branch offices around the world in order to be as close as possible to our partners and customers, as we can see here on the right. Sterilization by design is our identity. In our view, sterilization is not a regulatory checkbox or a process to be verified at the end of production. It's conceived and built upstream within the very design and management of the process. This is what sterilization by design truly means. Currently, about 50% of medical devices worldwide are sterilized using EtO. So the reason is clear, it works. EtO is effective for devices that cannot tolerate heat or moisture, and it penetrates complex geometries extremely well. But the strength also comes with a serious challenge. EtO residues and emissions are harmful to people and environment. And that's why we are a turning point. 2019, the United States Environmental Protection Agency, EPA, identified EtO as a significant health concern, citing cancer risk for workers and surrounding communities. This prompted the US Food and Drug Administration, FDA, to call upon industry stakeholders to actively seek alternative sterilization methods. These concerns have already led to the closure of commercial EtO sterilization facilities. The collective stance of the EPA and FDA indicates that EtO is no longer considered a suitable sterilization method without significant warning. Medical devices can be sterilized in several ways, including using moist heat, steam, dry heat, radiation, ethylene oxide gas, vaporized hydrogen peroxide, and other sterilization methods, for example, chloride dioxide gas, vaporized peracetic acid, and nitrogen dioxide. FDA has announced in January 2024 that it now considers vaporized hydrogen peroxide to be an established method of sterilization for medical devices, recognizing the modality's long history of safety and effectiveness. FDA recognition of H22 as a category A sterilization process, placing H22 on pair with moist heat, dry heat, and radiation. So here we see the categorized sterilization modalities, starting from the lowest one, which is the novel sterilization, and then established category B sterilization, and established category A sterilization. What means established category A method? Long history of safe and effective use, multiple sources of information like ample literature, clearance of 510 or approval of a PMA, satisfactory quality system inspections, voluntary consensus standard for development validation, and routine control that are recognized by the FDA. On the right, we see some example of established category A, like dry heat, moist heat or steam, ethylene oxide with devices in a fixed rigid chamber, radiation such as gamma, E-beam, X-ray, and vaporized hydrogen peroxide according to the ISO 22441. Fedegari, we are a global leader in steam sterilization for the pharmaceutical and biotech industries. But vaporized hydrogen peroxide is a technology embedded in Fedegari for more than 15 years, and more than 200 equipment already built, validated, and on production. So, sterilization using vaporized hydrogen peroxide has been introduced not to replace steam sterilization, but to solve problems raised with the use of EtO. So, the decision tree on the right has been developed to choose the proper sterilization method. When selecting a sterilization method, it's not just about killing microbes. We have to protect the essential quality attributes of the material, and we must achieve sterility assurance levels that meet or exceed regulatory expectation. Some key considerations include, first, intended use. We need to understand how the device interacts with a patient directly defines if the sterilization is required. For instance, devices contacting the bloodstream or other sterile area of the body, of course, require sterilization, while those merely touching uncompromised skin may only need validated cleaning and disinfection. And then product design and sensitivities. A detailed understanding of the device's material and design features is paramount. Factors like sensitivities to heat, moisture, chemicals, oxidation, and pressure change must be deeply evaluated. Manufacturing process like molding, chemical treatments can introduce residue, stress, or chemical that impact sterilization compatibility. For reusable devices, compatibility with multiple cleaning and sterilization cycles is also critical. So the selection of an appropriate sterilization modality is a critical step in medical devices development and ensures an effective sterility assurance process. An unoptical choice can lead to severe consequences, including inadequate sterilization, compromised device functionality, harm to patient, and costly delays in product launches and regulatory reviews. When the moist heat sterilization is possible, for sure is always the first choice. But when heat or moisture could damage the product, we need to consider low temperature alternatives. That's where hydrogen peroxide comes in. Here we see the milestone of hydrogen peroxide. It has been introduced in the market on 1991, where EtO2 was explored as a low temperature sterilization method. In the decade from 2000 to 2010, regulatory authorization were issued for industrial EtO2 sterilization application. In 2015, the US Pharmacopoeia recognized EtO2 as a sterilizant agent. In 2018, we already seen in the previous slides. ISO 22441 has been published and then recognized by FDA on 2022 and 2023. And last year, on 2024, FDA classified, as we already seen, EtO2 as a category A sterilization method. So the advantages of EtO2. So for sure, low temperature processing. EtO2 can sterilize effectively at temperatures as low as 40 degrees, with a typical operating range between 24 and 38 degrees. This significantly reduces thermal stress and potential damage to heat-sensitive materials that cannot tolerate traditional high-temperature sterilization method. And then broad spectrum effectiveness. EtO2 has high effectiveness against a wide range of microorganisms. When used for the sterilization process, it consistently achieves a minimum of 10-6 sterility assurance level. Environmental profile, EtO2 is considered a green sterilization modality because, sorry. Just a second, sorry. Okay, here we are. Back, EtO2 is considered green sterilization modality because its active vapor breaks down into oxygen and water vapor. This eliminates the dangerous emission associated with ethylene oxide. Excellent material compatibility. EtO2 has an excellent compatibility with a wide range of metal and polymeric materials. It's suitable for several products, including general surgical instruments, endoscopes, implants with electronics, pharmaceutical containers, and pre-filled syringe. Regulation validation as per ISO 22441 and FDA, as we already seen, approved for healthcare sterilization of medical devices. Despite its advantages, EtO2 also presents specific challenges that require careful consideration in process development. The first one is some material incompatibility. The primary challenge of EtO2 is incompatibility with cellulosic materials. Cellulose degrades hydrogen peroxide gas, compromising sterilization effectiveness. Additionally, certain uncoated reactive metals like copper and brass may react with H2O, leading to surface degradation or discoloration. These issues need careful material selection for devices and critically for primary packaging. And then some shape and complex geometries, direct contact needed, and secondary packaging constraints. In order to mitigate the challenge of H2O, particularly concerning penetration and residue management, deep vacuum processing is identified as a primary or preferred method to replace ethylene oxide and to improve the H2O penetration and the removal aeration. About the improved penetration, for complex devices, deep vacuum processing significantly increases the penetration by creating a pressure differential that facilitates the diffusion of hydrogen peroxide vapor into complex devices. Concerning to the efficient residue management, H2O offers a cleaner process compared to EtO, as its breakdown products are oxygen and water vapor. The application of a deep vacuum during or after the H2O cycle further improves the removal of any residual vapor and its breakdown products, and decreasing or eliminating the need for extensive post-sterilization aeration, typically associated with ethylene oxide. Let's compare EtO and H2O. So for sure, by environmental point of view, H2O is much more friendly than the EtO. H2O is not toxic, is not explosive. Both have high compatibility, as we already seen. The cycle time using H2O is much shorter than using ethylene oxide. And another important advantage using H2O is the quarantine is not needed with H2O. So, starting from this evaluation, we develop a H2O process, H2O sterilization process with the following process conditions. We work with a deep vacuum down to 1 millibar, with a temperature process between 30 and 60 degrees. The tailored process is based on the product features, with deep vacuum facilitating the H2O penetration and also the removal, the aeration as well. Some typical treated products are medical grade disposables like tubing sets, bullet lines, plastic or polymeric packaging, pre-filled syringe, sealed or semi-sealed containers, and complex shapes or materials sensitive to the steam. About the process, as we can see here, so the main phases of a typical H2O deep vacuum autoclave sterilization are first the autoclave preparation, means preheating process checks. The chamber is warmed up and the system conditions like vacuum H2O supply are checked. And then deep vacuum and loading condition. Air is evacuated from the chamber to achieve the deep vacuum, improving H2O distribution around the load. And then the third main phase is the H2O injection and sterilization. Vaporized hydrogen peroxide is introduced in one or more pulses, maintaining exposure to critical sterilization parameters for a set time. Sensors continuously track temperature and pressure to confirm uniform distribution. And then the post-sterilization aeration. So the chamber's ventilation reduces the residual of H2O in the chamber to safe levels, and a catalyzer is used to split hydrogen peroxide into water, vapor, and oxygen. So the question is, why limit your process to one technology? As Fedegari, we combine two technologies in one equipment. Means moist heat and H2O deep vacuum in one only equipment. One machine to suitable process to treat and to sterilize the widest range of products and packages. As we can see, moist heat, we can sterilize glassware, solid loads, metallics items, filters, textiles, stoppers in bag or no, wrapped items, and then all the items, materials that we already seen by H2 deep vacuum, like medical grade disposables, plastic or polymeric packaging, prefilled syringe, complex shapes, or heat sensitive materials. Okay, let's start with the first case study. Here we have saturated steam sterilization of ready-to-use primary containers cartridge product. The ready-to-fill manufacturing process in pharmaceuticals refers to the production of sterile containers that are delivered pre-sterilized and ready for instant drug filling. This approach is increasingly used in aseptic fill finish operation, especially for biologics, cell and gene therapies, and other sensitive injectable drugs. How we approach the study? So first, key parameters have been defined to preserve the product and the package integrity, in particular modulated depressurization and dry pulses. Then partial cycle and full cycle load evaluation of microbiological sterility effectiveness using biological indicators and checking the sterility of the product and the packages at the end of each cycle. Third step, the process release means cycle transfer for product process validation as per that ISO. So the initial evaluation has been done on the nest and tub in bags without the plastic bags, as we can see here on the slide, to understand the main potential wake points of the product, and then using the plastic box containing the nest and tub, as we can see here on the right. So concerning to the microbiological assessment, we place several BIs as follow. We place 7 biological indicators inside the plastic box between tabs and shown here on the left. 9 biological indicator on the right, 3 in the tab top left, 3 in the tab center middle, and 3 in the tab bottom right. So the critical quality attributes are physical, chemical, biological, or microbiological properties of the product that must be within a specific range to ensure the desired product quality, safety, and effectiveness. We identify for this specific product these following attributes, and some of these are impacted by steam sterilization, like package dryness, tub and nest deformation, shrinkage factor, seal integrity, and product sterility. For the process development challenge are here below listed and shown on the slide. First one, tray and nest shrinkage factor because it can cause dimensional change. Second one, lead stress anchorage is about ensuring the lid remains securely and fixed under mainly thermal stress. Third one, humidity degrees presence is important because humidity can affect, for example, material properties like plastic trays or nest can shrink in a different way under high humidity. Sterility and contamination risk, packaging integrity, especially for lids and seals. Fourth point, print anchorage stability measures the bond strength between the print and the substrate, like plastic tray, lid, or packaging material. It ensures that printed information like batch codes, barcodes, brand, and so on doesn't peel or fade during the sterilization. So the solution has been a process parameter modulation, working on the modulated depressurization and dry pulses, phase cycle adjustment, print stability, investigating different technologies like different direct print ink into the tub, print and sticks labels, and laser printing. Here we see the second case study that we want to share with you is the H2 deep vacuum sterilization of hemodialysis tubing sets. Hemodialysis tubing sets are extremely long and narrow, some up to 4 meters in length, with a 5 millimeters inner diameter, and not all vented. That makes the sterilization a huge challenge. Using H2 deep vacuum, we achieve uniform distribution, full sterility, and a total cycle time lower than 8 hours, even though that high complexity and without quarantine need. That's far shorter than a EtO process, which can take multiple days due to the aeration. On the right is the prototype built and available here in Fedegari, in our tech center in Italy, and where we perform all this study. How we approach the study? So we perform some tests, we check the biological indicators performance, and adjusting the key parameters like reducing or increasing the number of the pulses and the H2 concentration. At the end, we perform all these tests with the full load. The process is, as we already seen, composed by three main phases, which are the conditioning, sterilization. Sterilization means H2 injection, deep vacuum pulses, and aeration, which is a combination of deep vacuum and overpressure pulses. The results we obtain and we reach through the biological indicators verification a sterility assurance level of 10 minus 6, 12 spore log reduction, H2 residue lower than 1 ppm, and we checked if that deep vacuum effectively facilitate the penetration and the removal of H2 from the product. So currently, there is no any specific guidelines or regulation which fix a H2 residue inside the product. But just some, as we can see here, some general reference on the ISO 22441. To define these limits, manufacturers should perform a toxicological risk assessment in accordance with the ISO 1099317. Going back on this study about the hemodialysis tubing sets, H2 residues lower than 1 ppm was obtained applied this method. So several samples were filled with purified water and placed in an oven at 37 degrees. The extraction water was then analyzed by titration with permanganate to measure the amount of H2 extracted. That is not the only method on the market. Other ones are described in the pharmacopoeia, like that IVIS method. Okay, the third case study is the H2 deep vacuum sterilization for glass pre-filled syringe field. So this study investigates H2 sterilization of glass PFS filled with anophthalmic drug products enclosed within Tyvek plastic blister packaging. So the investigation focused on material compatibility like rubber plunger, blister, evaluation of the penetration efficiency through the secondary packaging, and the assessment of sterilization process effectiveness. Special attention was given to the assessment of H2 residue in the product and in the secondary packaging. So So here we see how we approach the study. The first phase was a small-scale study, defining the key process parameters, evaluating the microbiological effect with biological indicator, and measure the residual of H2O in the blister and in the product. The phase 2 full load study, optimizing the process in a full load or of PFS in blister, confirm the microbiological effectiveness and validate the H2O residues. The process parameters like pulses, H2O amount, vacuum, holding time were optimized to ensure sterilization efficacy and effective aeration. So the most representative cycles tested with an increasing quantity of H2O are here below. Cycle 1, 1 H2O pulse, total of 10 grams of H2O injected. Cycle 2, 2 H2O pulses with a total of H2O a total of 30 grams of H2O injected. Cycle 3, 3 H2O pulses, total of 60 grams of H2O injected. That was done to evaluate the efficacy of each cycle by microbiological point of view and the H2O residues. So the effect of the H2O, as we already mentioned, was evaluated by placing biological indicators in different locations. So position A, inside the syringe barrel lumen next to the plunger stopper. Position B, below the backstop flange. Position C and D, inside the blister toward the leading foil side. So with H2O deep vacuum sterilization, we achieve sterility, kept hydrogen peroxide residue below 1 ppm in the packaging, and ensure zero residue in the drug product. And what about the cycle time? Just 1 hour and a half. That show us that H2O can meet even stringent pharma standards while protecting delicate formulation. So these case studies highlight an important point. Developing an effective sterilization process is not about replacing one method with another one. It's about tailoring the process to the product by controlling injection, deep vacuum pulses, and aeration. We can in this way design H2O cycles that achieve sterility assurance while minimizing residue and protecting the material compatibility. Almost at the end, and here we see some conclusions. So, increasing the regulatory pressure on EtO use demands this transition to alternative sterilization methods that are safe, effective, and compliant with global standards. Choosing the appropriate sterilization method requires a systematic assessment of the product stability, material limitation, and regulatory requirements to maintain sterility assurance and product integrity. Vaporized hydrogen peroxide under deep vacuum is a reliable solution for complex and sensitive types of items, ensuring both microbiological effectiveness and material compatibility, offering an effective alternative to ethylene oxide sterilization. With its flexible process parameter, like we have seen, number of pulses, H2O amount, deep vacuum target, holding time, the deep vacuum H2O autoclave allows adaptation to ensure process effectiveness and optimization of the H2O sterilization cycle. So the transition from EtO to H2O is a critical challenge for sure, and an incredible opportunity at the same time that must be met head on. At Fedegari, we are committed to helping medical device manufacturers navigate this crucial shift with confidence. Developing an effective sterilization process is not about replacing one method with another, it's about tailoring the process to the product. So contact us and prepare your organization for this critical transition in sterilization.